is Professor Dieter Braun. He's professor at the Ludwig-Maximilian University in Munich. And actually he's not only a brilliant professor and is doing very nice work, but he's also a friend, because we met in Munich at the Max Planck Institute for Biochemistry. We were there together for a period in the department of Peter Frommerz, where he was working on characterizing mainly cell adhesion using optical techniques, and he contributed to develop original optical techniques for measuring cell adhesion. And then he moved to the States, and since then he's more involved in biotechnology and studying evolutionary biology. And particularly, I think now his talk will focus on particularly this latest aspect. He also will present something about techniques, I think, concerning new biotechnology techniques concerning measuring protein affinity. So I will leave him the word. So please.
Thanks very much. I try to entertain you as much as I can the evening. So please, if you have questions during the talk, things are unclear, raise your hand. We can have a dialogue type. It will be more entertaining. So I do experimental biophysics, and I'll expand it a little bit more towards the biological side. So I try to be an inspiration for you, try to show you a little bit what could be the connection, and think about what would be ways and strategy if you would go smaller and smaller and smaller. So the title is actually coming from the push to make the most shortest title ever, describing what we do. And of course, we are not doing Frankenstein, and we are not there yet, so perhaps make a question mark behind it. But I'll try to convince you that we have one way of pushing the limits towards driving evolution in an autonomous way, and I try to show you the recent advances in doing those directions. And I should start with the definition of life. I won't do that because it's hard to define, and I'll try to build on your understanding which you have acquired over the years. But basically what I'll have here is an E. coli, and the basic thing which you can see from this E. coli is that it's dividing. So those are micrometer-sized, could call it robots. They have the huge advantage that they can divide themselves. They have even more advantage, they can live off not, like a computer virus of computers which are running and therefore divide on those computers, but these creatures can really divide on simple chemicals. So they nourish themselves from very simple molecules. And actually the argument, if you would think about how you make a replicating robot, something which could replicate, I would argue that essentially you have to go down to a molecular level, because otherwise that's not how the world is structured. Your basic building blocks are not something larger, which had, coming from physics, a very low entropy state, which is so improbable that you won't find it, but you have to go for the smallest building blocks. So I'll have two tutorials in this talk to connect you a little bit to the topic, and both tutorials, one will be on polymers and biological side, and one will be on entropy. And I don't know, who of you knows, just to get an idea, who had the last time biological lessons in school and never heard of it later on? Ah, not too bad, okay. Who knows what's a polymerase chain reaction? Ah, wow, good, very good. Okay, so let's move on. So essentially, I'll try to convince you a little bit, or when we discuss life, living organisms, we have to discuss essentially evolution, because that's how life have evolved, have learned how to use the sequence space, this huge space of possibilities. And our work will focus on how can you come up with basically non-equilibrium boundary conditions to drive those evolutions. And we heard already that gene expression is slow. Yes, for sure, evolution will be also slow, and learning by evolution will be a very slow process. But there's some evolutionary process like our immune system, which is also evolutionary. It's very fast, We have evolutionary systems like our language, which is even faster. So, some of the concepts can be, abstracted and taken also to other levels. So it's about learning by evolution. I'll talk a lot about replication. I'll also talk about survival. Survival can be because you replicate well. It can be because you're fitter than the others, better matching. Many ways to think about that. And evolution actually made quite, I don't know whether you saw this movie before, very good robots which can even hunt down E. coli. So what you see here is actually a small bacteria cell. This is a white blood cell, and if you let them run, this is a nice tracking robot which can actually track this small creature down, which is not so easy because they've been optimized not to disguise themselves as much, and basically manages to eat it at the end. It's one of the strategies our immune system actually works. Now he got him. And, evolution has some real powerful mechanisms to come up with such solutions out of very difficult and very noisy systems. So our focus will be conditions for autonomous evolution. So we'll discuss a lot about selection, replication, because those are the hallmarks of Darwinian evolution, and how could you come up with strategies to implement that in the lab. Your experimental physics, only a very small part of what I will show you will be theoretical work. Most of it will be experimental work, because you really think that this is a big puzzle, and we only find pieces for the puzzles if we would go there experimentally and check what is the real working of these systems. Dobzhansky in 1964 said nothing in biology makes sense except in the light of evolution. So we also think if we want to learn something about evolution, better tackle it head on. What many of those talks we heard today, of course, work with things which have developed really late in evolution, humans, eukaryotes, more complex systems. The cells you just saw hunting down the bacteria has originated probably in this case 1.5 billion years ago. It's still quite modern eukaryotic cells, which is very large. And the first hint towards these bacterial cells, which we saw in the very first movie, actually goes 3.5 billion years ago. It's a little bit scarce, the evidence for that. But the interesting thing is actually that those creatures, they live off molecules, they replicate, they have all complexities they can divide. And those have emerged on the Earth right after the Earth was cool enough to have water. So it appears to be that, that fastness of that evolution, at least for me, is a motivation that we should dig a little bit more into that puzzle, because it might have experimentally tractable and possible solution, which is actually straightforward. I'll try to show you one scenario where we try to push that. Okay, so it's about early Earth and how to get that towards life, and I'll fill up these bubbles as we go through the talks, because we try to make a minimal autonomous evolution machine. So there are two things in it: minimal, right? We try to do it as much minimal small components possible, and autonomous. That's something which people are a little bit missing often when they talk about evolution, because they think about evolution, I'm in the lab and I'm mixing together my chemicals and I'm simulating the evolution. That's not what we want to do. We want to have it done by itself in a small system. And we go through this history, but I first need to make you aware a little bit about the players. So a short introduction, primer about biology, about RNA, DNA, and protein. So biology is all about polymers. That's something which, well, for biology it's normal, it's simple, but it's something to really memorize and to take seriously. So all the information which we store genetically is done in RNA, DNA, and those are encoding for proteins, and both are polymers. So we've got to deal with these these polymers and have to figure out a way how to manage those and make those evolve, because the evolution then will be the creation of longer and longer of those polymers, which have more and more diverse and more defined information and can do more and more interesting things. So what's an RNA molecule? Those are sugar backbone molecules with phosphates, and you have the 4 bases, as you probably will know. CGAU for the case of RNA, those are carrying the genes. So this is like the Turing machine. It's a memory of everything which is driving your cell. Those memory actually is not the only language. So here you have the genetic language. You have a second language, which is actually a second polymer, which is the amino acid on the right-hand side, which are proteins. Those proteins are made out of amino acids. Those are 20 of those. So you have to translate from the 4 ones on the left-hand side to the 20 on the right-hand side. And that's actually done with a large piece of RNA molecule, which actually connects with a 3-letter code down here. So 3 bases of that 4-letter code. And connect on the far other side towards the amino acids. So that molecule is actually made to make the transition from the genetic information towards the protein. Why is nature doing that? Well, you want to catalyze the reaction, you want to process those molecules, and those processes, some of them you can do with this RNA on the left-hand side, but it's been not a very efficient way to do it. You rather do it with the proteins because those amino acids are smaller building blocks, they're much more flexible, and with those you can do much more efficient reactions. So at some point, nature must have invented these 2 languages, and until now, never evidence has popped up that you could translate back from the proteins to the genetic code. It's not known that that machinery would ever exist. So it was a forward-feeding mechanism without backward information. There was a big argument in the evolutionary times of Darwin, that Darwin was saying, you know, it's not like the Lamarckian way you can have a protein which is getting better and better and therefore feedback on the genetic level. That's not what we're seeing. At least they're now popping up some methylation routes, but it's not the major theme. But you only have these random mutations on the left-hand side which feed into the proteins, nothing back. So those are the players. Basically, I discussed here what you can do from RNA to the protein. You have these tRNA you saw in the center, and that's done in a big complex called the ribosome, which also has a lot of RNA pieces. You now know, well, RNA is not the whole story. The RNA is actually memorized in the more stable form and DNA, and if you replicate your system, you actually replicate the DNA, and that replication is done with proteins. So you already have a difficult puzzle because you have these 2 languages, and obviously they're intertwined. So to make the proteins, you need the DNA, but to replicate and to translate that DNA into RNA, you need proteins. So you start to wonder what was first and how could you have those 2 things at the same time. I'll give you one idea of us, how we think we can solve that later in the talk. So that's the process in more detail, how you make out of these double-stranded DNA, these RNA, and that's this slow transcription we heard already from Joe's talk this morning, because the mechanism which initiates this RNA transcription and therefore production of the protein can be controlled by a lot of proteins on the left-hand side, and you can do really these programming. You probably know this Eigen competition, which is working a lot on that field. The second step is then, here's one cartoon of what I showed you before. You have this RNA coming here, you have these tRNA pieces here, which are connecting 3 of those bases with 1 amino acid, and then you can make those proteins. Okay, that's my rough primer so that you have some orientation. You have RNA, you have protein, and it's mostly, we start with the problem of how to replicate that DNA or RNA. So come back to our initial question, what conditions can we have for making this evolution in an autonomous way? How can we do selection and replication? So the machinery, there's a very nice animated movie by the Harvard University. So that's the robot for replicating DNA, which in every cell division has to be replicating the whole thing. So basically the double-stranded DNA comes in here. You have a number of proteins. One of them here, the blue one, is splitting it into both strands, and the one strand directly feeds into a polymerase, which makes the single-stranded DNA again double-stranded. So you have a first duplicate created here. The second one is a little bit more complicated because those molecules have never evolved to read it, to do that, that read in the opposite direction. So they first have to read it from the other side and then have to fill in the gaps as they go along. So that's the central Darwinian process. You want to replicate the DNA, and you see a lot of proteins. So you're again at this problem of how can you replicate that with proteins. And I'll focus a little bit on such a replication experiment because it has some details which already hint towards interesting boundary conditions. And those were experiments actually done in the 1960s, 67 by Spiegelman. He, for the first time, figured out how to purify those proteins, which you just saw. A little bit different ones. Those were replicating single-stranded RNA, but it doesn't matter for the thing I want to explain here. So you have a pot where you have these replicating protein. You also have some energy molecules. Basically, it's the single bases which you add towards single-stranded of DNA have the second strand, and therefore duplicating the DNA, RNA in this case. So you can plug in an RNA and wait for some time. And what he was then pursuing was, okay, we check out what's happening if we do this over and over and over. This over and over means that from an initial reaction after some time, I take a part of it, but only a small part of it, put it in the exactly same tube, again with new protein, with new energy-rich molecules, to have another replication. So that's an evolutionary cycle. You have a replication going on, and you have a selection. So selection, we'll come back to that, because this experiment resulted in an interesting problem, actually. So he did that over many times, and checked what the outcoming RNA looked like. And at some point, this was called the Spiegelman Monster, which came out of this. But essentially, it was an astonishing result because you started with a long RNA sequence, 200 bases, and after already 30 of those serial dilutions, as it's called in the literature, he only get very short RNA piece. So apparently, the selection pressure he was putting by just doing a dilution, and that's, that's typically what you would expect for your creature to be dispersed by diffusion, other things, that's a serial dilution, he gets shorter information content. So the information gets shorter and shorter. Basically, what these RNA molecules at the end were representing was that only that protein can still bind on it, but nothing else. So these RNA, which were initially coding for virus, were really aggressive for E. coli, they completely lost their function. So it was not a monster, but rather a fly, which it could evolve with such a mechanism. And the reason is actually very simple. So if you think about if this polymerase just docking on any RNA molecule floating around and duplicating it, shorter sequences will be replicated much faster. It's about 6 times faster per time unit. So if you have the same time you want to replicate 200 bases, you replicate, and you have then a factor of 1, 2, 4, 8 for that replication. In comparison, the 30 bases go like 1, 6, 36, 216, 1,200 something, right? So it's a huge advantage of those short pieces to replicate. So how can you come by that big problem of how to replicate rather the large pieces? Because at the end, you want to have an evolution which creates more information, which creates more structure. So how can you replicate long DNA? And we have a way where we think this is helping, which comes a little bit unexpectedly. And it's basically actually to drive a replication reaction with thermal convection. So the basic reaction I'm pinpointing to is actually well known because we are hitchhiking a little bit a method from biology here, which is a Taq polymerase. And that has the interesting aspect of the following thing that you saw in the movie before, that you have this replicase which actually has to split the DNA in 2 halves. That's a rather complex issue. You need a special protein to do that. So this reaction is solving that just by heating. So you heat the whole solution to 95 degrees, you cool it down, and have then these green dots, the polymerase proteins, attached to, in this case, special starting points, and replicate from those things. The big advantage is this, of this way to do replication is now first of all, you have this separation at the high temperatures, so you solve that. You need one protein less, makes life much easier. And the second thing is that even if you have long or short DNA, they replicate the same speed, because it has to wait until next time, so this is cycled, of course, from here up to 95 degrees, next time you get a high temperature step again. So your temperature stepping is actually gives you the speed, and you have a good chance that your Spiegelman fly and your Spiegelman monster is both replicating at least at the same speed. So that's helping a lot if you want to come up with a Darwinian scheme where the short pieces, which are crap, have no information, are not outcompeting your long ones. How that works out, things I did postdoc time, is actually you only need to have that liquid and heat it at one point. What's happening in such a liquid is actually that you heat it, you have lower density of the liquid there, so it moves up. And as those proteins, that mixture goes along with those convection flow and all this laminar, because things are small, you basically apply a temperature oscillation. I have a movie of this if you look from top, and if I'm allowed to, on it, that's how it cycles. That's about real time. So things are cycling from hot, cold, hot, cold, hot, cold. So that reaction in the center, in this hot center, is melting, the double-stranded, the single-stranded, and outside here it's replicating in the double-stranded again. So that's nice because you just, by applying this temperature gradient, you can have a replication reaction, and the full analysis needs fluorescence detection of those species, and if you wait for a while, you see that this double-stranded DNA comes up. That's this increase of fluorescence you can see here. So the detailed analysis actually shows you that the DNA is indeed doubling every time, and it does double for the short ones or for the large ones the same speed. So that thermal convection gives you already a very important solution to implementing that replication reaction towards having all replicate the same speed, because otherwise the simple ones will outcompete the large ones, and you have rather degradation of information and a build-up of information. So here we don't yet have a build-up of information, it's just equal par, so they are at the same speed. Okay, that's first part. What I tried to show you is that if you want to make a replication selection reaction, take care that your replication is not selecting already for the poor species, but is helping actually the large ones also be replicated. And one way to do that is such a thermal convection flow. We'll learn in the second part of the talk that this thermal convection has a second aspect which actually increases the concentration of the constituents at the same time. So, but it's time for second break and second recapitulation of important stuff, because I've got to focus now that I told you here that we have a temperature gradient. You can say, okay, well, we need that for this convection flow, but I'd like to argue that it's exactly essential if you want to build any Darwinian machine, any evolving machine, that you have to think of any non-equilibrium driving of what you're doing. And that's based on the second law of thermodynamics, and that has to do with entropy. So that will be absolutely the same if you have robots to replicate, if you have other things which really go under evolution. You'll have some battery, you'll have some, you name it, energy source driving to make these things happen. Otherwise, it won't work. So what's entropy? Very confusing concept sometimes. So basically, this plot explains more or less everything what you need to know about entropy. You have configurations of your system, and those configurations, you can count them, how many of those configurations you have by multiplicity, and that function has a strong peak. So that's for typical systems, for the air here, it means that the air is typically, diluted everywhere, and you don't find all the air molecules in one corner. It's not because they want it or they don't want it, but it's a simple thing of quantum mechanics. You have the states, you calculate how many of those states you have, and you calculate how many of those have the same equal distribution, and you find much more of those in the center that everything is distributed than those on the right-hand side, they're not. So a typical curve of this is very extremely peaked. So you have one peak, and then those x-axis actually you can wrap around the Earth more or less, and you get a very low number of those configurations. So it's a probability to have the system in these low number of configurations very low. So basically what's equilibration? If you start things out of equilibrium, they equilibrate into equilibrium. You basically just increase the number of configurations. So the entropy is going up as you approach your equilibrium. That's something we don't want to have because, for example, this means that out of a highly concentrated pool of molecules, you end up with a highly diluted one. Well, the highly diluted one will not react interestingly, doesn't make interesting things, especially the very same argument you can apply for these polymers I told you. So all life is polymers, and you see already, well, those polymers are this small tail of low concentration, low probability on the right-hand side. So you don't find those configurations of those polymers. So how can we, push the system to go from this boring equilibrium state towards those disequilibrium states? Well, typically only for a very small system, this entropy is decreasing. The general law of thermodynamics tells you that the entropy is going up, which is just meaning that you have more states to share those configurations. So we need to push it out of the equilibrium, and one of the ways to do that is actually to have a small system, denoted here with the red corner, which is actually able to decrease the entropy in a system where at the same time the whole system is increasing the entropy. So the whole system is increasing the entropy, but only a small system is allowed to decrease it. That's exactly the situations I'll discuss, and that's what you need to run a Darwinian evolving system. If you don't have that, you will never reach these interesting, highly evolved situations. So it's already indicated that we discuss heat and heat transfer. So basically, the net entropy generation of that heat transfer is, for the total system, positive. But as I'll show you, you can create small systems where we have this heat flow going through, where the local entropy of that system is actually going down. So we can actually use a temperature gradient not only to convect that liquid, but also to reduce the entropy. So out of monomers make polymers and make high concentrations. So that sounds a little bit like magic, but I show you the exact mechanism how you can do that in a second. Probably won't go too much into this. So basically, when people are discussing evolution, they don't really take care of this fact well enough, in my opinion. So they have high concentration liquids, they put it together, let it equilibrate. They put, a single-stranded DNA, put some second single-stranded on it, let it equilibrate. They have things on a clean surface, put it on the surface, let it equilibrate. They put things in a membrane, let it equilibrate. So it's never a thing which is driven out of equilibrium, but people are rather studying dying systems as they equilibrate. They're not studying living systems in an equilibrium. And those are quite artificially made systems, and it's very hard for those cases to think of, non-equilibrium systems which can do that, right? So how you can bring things into membrane? Well, in the experiment, of course, you start with high concentrations, you make a membrane, you dilute, you get rid of the high concentration outside, then you have it packaged inside, but of course they will degrade, they will get out, so all the mechanisms which now make those bags of highly concentrated molecules are, of course, membrane proteins, highly evolved proteins, which pump those things all the time into the cell. And it's hard to imagine that has been done in the very first steps, because those proteins are really rather late development. So we want to go for real equilibrium, in our case the thermal gradient. You can imagine other things, but there must be some disequilibrium system. At best it should be permanently driving the system all the time. We'll focus on thermal gradients. I'll show you that I think we can do at least some more experiments in that. I'll show you where we are. So the first thing is the concentration problem. So I told you, how can you get enough molecules? Because you want to have them crowded like in a cell. You want to have them real reacting fast, doing polymerization reactions, to push them from diluted ocean conditions. The ocean is very diluted nowadays. For sure it was, according to geologists, also 3.5 billion years ago. That's just, you go from timescales of microseconds to seconds. The complexes dissociate at lower concentrations, and you want to really push it for high concentrations. And there's some effect which we came about experimentally, which actually is helping this. And it's not so well known. It's a movement of molecules in a temperature gradient. And actually the basis of one biotech company is from two of my PhD students. But we'll focus on the basic science first for this. So basically, molecules do have a tendency to go from typically hot to the cold side. That's called thermophoresis. It's based on the energetics of the molecule, and you can record that in the experiment. So you have an infrared laser which heats the center by about 5 Kelvin, and you see how the molecules spread apart. If you switch off the laser, the temperature equilibrates very fast, and the diffusion equilibrates a little bit slower. So that's a basic effect. You see that this darkness of the spot is about 10%, 20% of concentration. And okay, it's a small, in German you would say dirt effect. So what do you do with it? It's not doing anything, and very much of it. But I'll tell you that this small effect can be fundamentally amplified, and actually out of a depletion where molecules want to go away from the heat, actually be used to concentrate those molecules. So please bear with me to explain that. We have a model to describe it. Basically, it says that it's the shielding, the ionic shielding on the surface of the molecules, which are driving those molecules from hot to cold. Those are experimental tests. All what you need to memorize here is that the larger the molecule is, the more they go along the temperature gradient. So not only that we move molecules, we move the large molecules much better. Remember, we wanted to have a good selection strategy for larger molecules, so that looks already interesting. So how can we make a trap out of it? I was proposing to you that if we apply temperature difference, warm and cold, that I get this accumulation of the molecules. And yes, you do get it. I'll show you experiments in a second. And it's a combination of two effects. Basically, the convection you saw before, now not in a flat chamber, but in a rather elongated chamber. And you have this thermophoresis, this movement of the molecules from hot to cold. So that combination, if you have the right size of the chamber, the right speed of the convection, typically those chambers have a width of 200 microns, gives you a very strong accumulation of those molecules. And because we have measured all the diffusion coefficient and these properties in the temperature gradient, we can actually make predictions. The predictions tell you that even small monomers, nucleotides of RNA, for example, accumulate very strongly in such conditions. So you may wonder where we get the temperature gradient of. That's one way which is contemporary now in the ocean. You get highly hot water springs on the ocean. It's a hydrothermal vent. There are two cases nowadays. In the early Earth, those structures probably looked different, but at the same time, you still had quite strong temperature gradients. Those are highly porous rocks because they're volcanic rocks. Similarly applies for very early Earth. So you have these pore spaces. Inside these pore spaces, these temperature gradients, and these you can accumulate the molecules. And that's a theoretical calculation, what you expect from these molecules. So if you have a single base molecule, you have to have a large aspect ratio. Things have to be rather long to accumulate. But as you see on the left-hand side, you get huge concentration factors, and they get huge and larger if you have larger DNA molecules. You can have basically an analytical formula for that experimental effect, which you can test, and gives you a very strong accumulation of larger DNA. So that's interesting because out of those replication reactions, you want to select the larger molecules the best. So is that pure theory or not? No, it's not. So this is an experimental run for quite inefficient trap. So this accumulates tenfold 5,000 base, rather large DNA molecule. We have come up with a more efficient way to do that. Basically, what you do is that you have to make that chamber a little bit longer, and the aspect ratio has to be larger. Without going in much in the details how this is done, this is accumulating here 40 nanometer beads. It's driven optically, so you can move around and actually have all those molecules come together at very high concentration in the center. So those are beads. Optical trapping can't provide you this high concentration. And if you go for 5-base DNA, a very short DNA molecule, you get 100-fold accumulation. So yes, so the mechanism is there. So out of this small depletion with this convection, you can get these high concentrations, and helps you a lot in getting your reaction going. Basically, Jack Shostak, 2009, picked up on the idea, and he inserted one of his lipids. And those lipids were brought into such chambers at concentrations below where they can form vesicles, and the concentration essentially brought them above the threshold of vesicle formation. You get vesicles. So if you are interested in making vesicles, you also get those in these reaction chambers. So I told you that just applying a temperature gives you high concentration of your molecules. That's interesting, because out of that you can probably do also some polymerization. And here comes the second part of it. You want to have those long polymers. You want to have those long RNA molecules, DNA molecules. Can this thermal trap also enhance polymerization? So that's the question. So we have this thermal trap on the left-hand side here. You have monomers. What's happening? Do you push it towards having those polymerize, and those traps enhancing that reaction? So we model that reaction, and that's first now a theoretical treatment of the problem. So basically what you normally have, and we tuned the initial conditions such that you don't have much of that polymerization. So without the trap, you actually only have this small piece at very low concentration and very short RNA polymers. So you basically have monomers in your reaction chamber. If you now switch on the trap, you get, after a reasonable time, very long polymers up to the length of 70 bases. So the trap is concentrated, it's concentrating, and actually can work against that polymerization reaction in a way that you get very long polymers. That's what people would love to have. Large RNA polymers, they're reactive, they do catalysis, they do interesting stuff. The same point I show in the left-hand side, it's essentially the total concentration of what you're enhancing goes up exponential with the trap height. Actually, it's going hyper-exponential, as the theory is showing us, so that you really get very high and very long polymers. So we can make a phase map of that, how it's really behaving. We can do the full theory. Have two limits of depending on whether the polymerization is fast or slow versus the concentration, and rather well understand the system, because we also did an experiment. The experiment is not yet having a real polymerization base by base on RNA, but it's using sticky pieces of DNA up here, which you can control optically. And I won't go through this busy slide. Basically, it confirms the theory and tells you that those approximations we took when calculating the theory are correct. So with this, you can actually make that transition I told you for this high entropy equilibrium state into these configurations, which are very scarce, but which are very interesting for evolution. And all you do is apply temperature gradient, nothing more. It's very basic non-equilibrium driving. Okay, so the question is now, I showed you in the very first part of the talk that we have the replication going in a thermal convection, hot, cold, hot, cold. Now I showed you in the second part that those traps are accumulating material. You might wonder, can't we combine those two? Because basically what you'd like to have is have the selection with the molecule trap I'm telling you all the time that those traps are better for larger molecules, so you make more complex stuff and it's trapped there better. And have a replication which is helped by the convection, because the large ones are replicating at the same speed, and the large ones actually also can separate by that temperature oscillation. So in the high temperature, you separate the DNA. And I wouldn't tell you that if we wouldn't have done the experiment. So you can really combine those and it works. So you can have a configuration where you have a trap and you have the replication at the same time. A lot of details, so we drive it with laser to be a little bit more defined with our temperature gradients. And basically that's the end result. So here we look with fluorescence what we have as replicated DNA molecules. Those replicated DNA molecules will show up at high fluorescence intensity. But in normal replication, as you saw before, it lights up everywhere. The dark spot we had initially is only for the higher temperature, therefore the dye is not attached anymore. But here in this trap, you really see at the same time, after a while of replication, to get beyond background, that those molecules are only accumulated in the center. So the trap is actually much longer. It's about 20-fold longer to be able to do that. Again, we need high aspect ratios. But basically, you can have the same time replication and the selection of the molecules. You do theory to understand what's going on. And we are heavily working on this to do the following, to really show that Darwin process, namely to show we do a short DNA in it and we do a large DNA in it. And the calculations predict that the large ones will survive, will have a much better replication than the short ones. And then we have an interesting machinery because it can replicate, it can actively select and compact the results against diffusion, and it will conserve the large, interesting molecules against the short ones. And then I think we will be a little bit close to, depending on how you define life, to have something which is really autonomously evolving, and that machinery should yield interesting results because we don't know yet what will come out of that machine. We tricked, of course, a bit. We tricked in the point that I used a protein to do the replication. So I told you in the very initial slides on the biology part that you have to have, how do you solve that chicken and egg problem? That's called the Eigen paradox, exactly. So how can you have that RNA molecules code for proteins, but need for the replication against proteins? So this is getting a little bit into biology, so I'll try to hop over it a little bit. But essentially, we have an idea how, and tested it experimentally to solve that one. Again, I told you those DNA or also RNA replicating proteins I showed you in the experiments are proteins. So how you can do that if you need it for encoding for the proteins. And these are the complexes for this translation from RNA to the protein. It's huge complex. Nature can do similar reactions like this, replication of DNA with much smaller proteins, very small things. And this machinery to make proteins, it's enormously complex and enormously inefficient. As compared to DNA polymerization, this is very precise, 10 to the minus 8, 10 to the minus 9 error rates. This is 10 to the minus 3, it makes a lot of errors, it's very sloppy, it's very slow. And mostly, I guess, that's bad reaction of that whole complex is because you have this huge molecule. So remember, this is this tRNA which connects this 4-letter code, you need 3 letters of those, to code for the 20 letters of the protein code. And you may ask, well, why do you have these, fancy, huge molecule? Why not attach that amino acid very closely down here and have a much more efficient way to do it? And, our argument towards that is that it's done in a way that this molecule was, in the initial way, a very basic replicator for RNA sequences. So that the tRNA was actually used as a replication device. People are discussing a lot RNA molecules, how RNA molecules can polymerize themselves. Those are quite large molecules. They have a huge disadvantage that they have to run at very high salt concentrations where they're degraded actually themselves at the same time. And it's not really clear how, you know, those machines would connect to the biology. It's rather missing links, and we'd like to make a concept where we actually use those tRNA molecules for replication. And bear with me, I try to make it as short as possible. Basically, what this tRNA molecule has an interesting aspect. If you do 3 mutations on it, it folds into 2 double hairpins. So this cloverleaf structure you see on the top side is actually combined into 2 double hairpins. And we just take 1/2 of those double hairpins and made a replicator out of it. That replicator is never replicating base by base, as those proteins can do, but it will replicate those short sequences here. You know, remember, we anyway need 3 bases to encode it, so we need a little bit more for this scheme, so it's 9 bases of those, but we are mostly replicating those codons, those protein encoding pieces. And the interesting part of it, we don't need any chemistry. It's a very fast process, and we only need temperature oscillation again. We also don't, the whole energy of the reaction is driven actually by keeping the whole reaction at high temperature initially, cool it down. You can imagine that also being done by convection. Then you have those 2 hairpins, and they only react if they find a matching sequence where A comes before B. So the whole thing, what it memorizes, what it replicates, the information of which sequence is before which sequence. Here it's A before B. If you have that duplex, let it equilibrate at 10 degrees Celsius for several seconds, you get this conformation. And this is here very stable, so the hairpins are opening up, they hybridize to themselves or make a very stable stem, but they're very unstable towards this AB sequence. So if you heat it to 40 degrees, they separate again into 2 pieces, left and right. Both pieces replicate again with 2 hairpins. It will be an exponential scheme, and you can replicate and replicate and replicate. So that replicator uses this useless tRNA molecule in a very efficient way, and is actually very selective. You can have on the gels all the protocols to see that you have those products. You can confirm that it is a replicator. You can even confirm that it has, to a large extent, an exponential characteristics to it. It has a false positive rate, which is creating those structures by themselves at no sequence AB, but it has also an exponential part to it. So what's the idea behind that? It's the following, and those experiments we're performing right now is that if you have these 3 mutations from this cloverleaf to this double hairpin, that you can imagine those hairpins are not connecting now AB sequences, but A, B, C, D, E, F, G, longer pieces. And that's exactly what you need for this translation from this 4-letter code into the protein code, right? And those double hairpins are likely to produce these sequences and replicate that sequences. So now imagine those hairpins, initially this tRNA is really complex because it has this amino acid up here and the coding sequence down here. With the work of Michael Yauros, it's coming more and more clear that those amino acids which are up here actually want to bind down here. But in this structure, it's completely unclear. If we make these mutations, that binding site is down here, can nicely connect us to those codons, and you have a direct way how to connect those amino acids to it. If you replicate it now from those structures, you have all these amino acids up here, reasonably close together, and there are a lot of interesting RNA sequences which can actually connect those amino acids at physiological concentrations, very fast, very efficient. So RNA is a really good way to do that chemical reaction. So our proposal is that you have these tRNA to replicate those sequences and then have a starting scaffold for the translation. Experts know the work of Kuhn and Vaser, so we kind of invert that technique and have tRNA going. So can we have a way to select for those tRNA replicators? I showed you that you have these large sequences with these trap. Yes, well, that's a theoretical thing which I'll probably go over quite fast. It's well known chemically that those reactions actually degrade by cleaving much more if they're single-stranded and double-stranded, and you can show that actually you almost get a replication reaction out of that selective degradation, and out of that effect, we think that we can generate enough tRNA sequence to have these tRNA molecules around. Okay, so I'm at the point to wrap it up. So I tried to convince you that this convection PCR brings the replication for short and large pieces to the same level. I showed you a thermomolecule trap, which can trap the monomers, but also can selectively push the polymerization. And combined with both, you can actually have a replication trap, have replication and selection at the same time. And although we're using here proteins, we have some idea on how to get replicators also just out of tRNA molecules and have a better understanding why this tRNA molecule is so large and complex. So those are things we're working on, especially this Darwin trap, we call it, survival of the longest, but some other aspects. And that's our puzzle as we see it right now. We have this encapsulation in the pores, out of equilibrium with the temperature gradient. We have the molecules. Those are concentrated, forming polymers. We have a way to replicate those, either with the polymerase, with the protein, or also without, and have some ideas of how that can get into a Darwinian evolution scheme. So we have an ERC starting grant for that, running for 2 years more. So we also do other things, kinetics in living cells, light-driven microfluidics. I have these two were my first PhD students, which are actually using this movement in the temperature gradient to detect binding affinities, and they're doing extremely well. They have now 10 people in the company, and a lot of interesting biological affinities have been published. These are the papers from the last year, so they're selling the instrument quite nicely. And I'm at the end and try to, and those are students, the first row, the students right now in the lab, the last row, some of them biotech, some in academia, quite a bit in academia, like all my teachers where I learned science from. Thank you for your attention. Questions? Yes.
Thank you. So, one curiosity: you talked about replication of DNA, but would it be possible to use the same approach for transcription, for example?
At this transcription, for transcription, you first need the differentiation between RNA and DNA. And from what all we know, those molecules which are doing the transcription, I mean, basically this protein is a rather late development. So you're not really discussing that those steps were central in the first steps. Also, this polymerized protein back here was actually invented twice. And, oops. That's bad. Why don't we have any? Yeah, sorry. Somehow we don't have juice here. No current. So basically, can we have some? So basically, that step from DNA to RNA is something which has evolved later on. Once you had the translation to the proteins, you were developing that later. That's a little bit the consensus of the field, that you did that later on. So sorry, my computer battery is out. Yeah.
So Dieter, I wanted to ask you about the trapping process. Is it true that that needs to be a very specific size range, do you think, for this convection mechanism to start trapping? And, is that something you imagine just being generally available in the early environment? Is it particles of silica or something like that?
So it depends on the width of that channel, whether you accumulate large or small pieces, but that depends, is very small. So if you have about 100 microns diameter, you're selecting out the real long pieces, like 1,000 base pairs and longer. And if you have that 150 microns, not very much different, you accumulate the small molecules. So it is depending on the diffusion coefficient, but only to a very small extent. I think the third square root, the third root of the diffusion coefficient, defining you that. So still, those chambers, all the simulations I showed you was for the polymerization is 10 degrees, which you need across those 150 microns. So the gradient is a little bit on the steep side, but the geologists, also these, it's a good evidence that you rather have the temperature gradient across the water because it's not so much thermally conductive and not so much in these rock samples. So there's some aspects that you can concentrate those temperature gradients in the chambers. And those are very porous rocks, right? You have all kind of conditions. If you have the right ones, you accumulate it. You don't need to have that accumulation in that very defined chamber. It can be broken. Diffusion can overcome it. You can accumulate big bags of those molecules. You have many ways, and I think we showed it in very different geometries, a quite robust way to accumulate those molecules.
I'm curious about the future of this line of research. So what do you want to do after you obtain long genetic molecules, for example?
So the nice thing on all these experiments, I mean, they developed a little bit as we went along. So we were not designing this research to be, where it is now. Now we know a little bit better. We like to push. The interesting thing we don't know, you have an evolutionary experiment. It can, create all kinds of stuff. So you can select for length. You can also try to select for sequence because you can offer a sequence with a very long DNA. So it will bind to that sequence and therefore be accumulated. So that's one thing. Something I didn't talk about, we see if the DNA can actually, if you're not polymerizing small pieces but larger pieces, they actually form round spherical objects. And what we see under those conditions where you have that, you get big clumps of DNA, 50 micron large, possibly because all those rings are interlinked. And therefore you have a completely new level of how it's a kind of a solid state DNA, something we were never expecting. But that's a little bit what we are after. The physics people know if you're out of equilibrium, a lot of structure will come up, and we'll try to measure those structures in as broad a range as possible, but also a little bit focused, of course, towards the Darwinian evolution process. So let's see. I mean, for example, that tRNA double hairpin, that in a trap, how does it behave? We have some ideas, but it might be a very interesting replicator and provide quite large range structures. Let's see.
Well, I'm curious about the length of the DNA. I mean, if my recollection from biology is correct, in the DNA polymerase has a ring which is capable to keep the two strands of the DNA attached. Otherwise, the binding is too weak. The hydrogen binding is too weak in order to keep a long molecule.
You mean the polymerase now?
Yes, exactly, okay. Yeah, the polymerase. So I wonder whether in the case of your replication thermal, let's say in your thermal system, yeah, whether this is a limitation in order to reach very lengthy molecules or not.
So on the one hand side, the TAC polymerase, which is done for these PCR reactions, they do work very well at 70 degrees. They do it, they replicate, like 5,000 bases, no problem. So the temperature tuning for this tRNA replication is a little bit more tricky. So for the two bases, we need to really go to 40 degrees because at 40 degrees where it separates, at 10 degrees to react. But if we now do the double hairpin thing, we are probably pushing that temperature range to higher temperatures. And, depending on the reaction, you have to tune a little bit the temperature. But, that's an open parameter. We accumulate the same way, do it at 0 degrees or at 80 degrees, at 80 a little bit better, but you only need the temperature gradient. The gradient is pushing your concentration, pushing your replication, but it's nothing, we don't care at which temperature you're doing that. And basically, if you have a large-scale temperature gradient, you will have at the same location those different levels of temperature. Those will have diffusive connections, and it's a big playground of those structures and those convection cells. So that's another thing to think about, have different of those cells. One is optimizing to make the hairpins, the other one is optimizing for trapping, the third one for polymerization. Connect those, think of all the different ways of evolution which you can drive those. We're running out of power everywhere.
So, thank you very much.
Thanks.
The talks for the day, we have a discussion, and I'd like to invite the plenary speakers from all 3 days to come up to the stage and be part of the discussion panel. I'm not sure how many of you are still here, but if you're here, if you could come up, we have chairs. For those of you who are worried about breaking into your evening, the plan is to finish by 6:45, and there's still time then if you want to do the La Pedrera tour. And of course, Barcelona is really only just waking up by 8 o'clock, and there'll be restaurants going all evening. So I want to just thank again all the speakers from today. It's been a really fantastic first day, and I think we should congratulate ourselves. Thank you. So if the plenary speakers could come up, and also a couple of other people. Frank, you said you'd help, and Mark, and David. David, you stood up. That should do. I think that should be enough people. Can we have the screen back, or is it going to be a problem? It's okay. It doesn't matter. We can go from here. So I have some questions on the slides here. I have just 3 slides, and there's about 2 questions on each of them, so don't be intimidated. We've planned two of these sort of panel-like discussions. So today we wanted to discuss general questions around biomimetics, and tomorrow we are going to discuss the future of the field and what work we think is important or interesting to do in the future. And Nathan Lepora, can you wave your hand, Nathan? Has been preparing a roadmap on biomimetics research for the Convergent Science Network, and we have a draft of this already. We're hoping to publish it, and we plan also to distribute it widely, including to influential people like those that write European funding calls. So that's for tomorrow. For today, since this was the first time that we have run this conference, and we're very hopeful after getting a positive evaluation of our proposal for another coordination action that this won't be the one and only Living Machines meeting. So we wanted in this discussion to talk more generally around biomimetics and what we mean by it, and what we might do as a community. So the first question was, why copy nature? Two general reasons why we might want to copy nature that we mentioned in the preface we wrote to the proceedings is in order to build technologies that could be useful and would solve unsolved challenges that we have in science and engineering. And the other reason why we might do biomimetics is to better understand nature itself. But other people might have their own reasons or their own interpretations of those reasons. So I'd like each of the panelists to comment on why we copy nature, and perhaps also say when we should use biomimetic approaches and when we should not use them because they may not be the most appropriate approach. So if we start at this end, and Joe, if you could comment first.
Well, early on in my career, I got very interested in the question of whether the mechanisms that occur in the nervous system when studied in vitro are in fact the mechanisms that occur in behaving animals. So it's because the nervous system is under such heavy modulation by circulating neuromodulators, etc., when you study the isolated ganglia in a dish, there's no guarantee that in fact the same mechanisms are in operation as they are in the behaving animal. And this turns out to be very, very difficult to do, and extremely difficult to do from the right neurons, because often the really, really important neurons are the smallest ones and the technically most difficult ones to record from. So my primary motivation in doing biomimetics when I started was to really develop embodied simulations of how the nervous system operates, so we could begin to address this idea of what are the mechanisms in the operation of these closed circuits. So that was what I was after.
Yeah, I think similarly along the lines of synthesis. So probably everybody is aware of the famous Penrose Triangle. So you can draw a triangular structure that when you look at the three vertices and split them apart, these each make sense in themselves. But when you try to put it together, it doesn't make any sense at all. And so biology can be a lot like that, right? So in the sense that in the struggle in the European Renaissance between Descartes and Spinoza and other people like Giambattista, basically Descartes won, which was that we should be reductionist and basically search for greater and greater and greater details, and that somehow this is going to answer everything. But of course, that's not the full story, as the Penrose Triangle shows us, that we can understand the vertices and the pieces in isolation, but when we try to put them together, we don't necessarily get a system that works. And so these biomimetic approaches are very interesting and exciting to me, because they're one of the few examples in science, engineering, technology where you really get to test these principles directly, and you basically you either end up with function or you end up with nonsense, and this all depends, of course, in what's selected in the answer of what you think is important.
I just realized I didn't introduce the whole panel, and not everybody has met everyone else yet. So just to say who everyone is, so Joseph Ayres, we've already heard from today. Tim Pierce will talk tomorrow, and Toshio Fukuda. David Lane, who works on biomimetic systems, partly for underwater robots. Barry Trimmer, who will be speaking tomorrow. David Lentink and Dieter Braun, we've heard. Frank Grasso, who works on invertebrate neurobiology, octopus and the like. And Mark Kwiatkowski, who works on StickyBot, climbing robots and hands and all sorts of other things. So, and then just another thing to say is that we've got roughly 10 minutes per slide, so we've got about 1 minute each for this discussion, and allowing something from the floor as well.
Why we copy nature? Maybe we can learn from the nature in terms of such a kind of morphology or functions. so that we can extract those kind of shape or in the geometrical shape or structures, as well as such a kind of function. So we can make something, new device like that, which is a kind of efficient, energy efficient, or also then we can go in more in detail, such a kind of sensing, kind of control, and also structure ways, so that today many kind of two panel speakers already mentioned about such a kind of neural-based control or something like such a kind of protein-based things. So eventually, maybe we can have a hybrid system such that from such a inorganic material to the such organic system, which means in protein. That's a kind of interesting topics.
I think the reason to study biomimetic systems is because we learn things that help us build and engineer systems better than we can at the moment. I'm going to give you two examples. I went off to look at where the word biomimetic came from. Maybe you all know this already, but I didn't until recently. The word biomimetic was coined by a guy called Otto Schmidt in the 1950s. He's the guy that came up with the idea of the Schmidt trigger, the electronic circuit. He was studying, I think it was octopus or squid at the time, trying to look at the neurological structures and build similar structures. He built the Schmidt trigger, designed it. It's the thing that sits behind a flip-flop, which is a digital logic gate, which is at the heart of a register and a counter, from which we get digital electronics. So I think that the roots of digital electronics go back to biomimetics, right? That's just one field that's come from biomimetic studies and the inspiration of doing that, right? In my own rather lesser world, we work on underwater stuff, underwater robotics. We spent 10 years working with primarily US Navy, looking at marine mammals and acoustics of marine mammals. We're just starting a company. It's now 9 months old, called Hydroson. I'll give it a plug. Right, we're building wideband sonars using multiple chirps, which is exactly what dolphins are doing inside the click structures. And we're having great success at characterizing objects in the ocean that we haven't been able to do previously. And we believe we have market advantage and all those good things. So to coin a phrase that I first came across with iRobot, the people who make, which is their mantra of their company, the guys who make the little Roomba house cleaning robot, their objective in life is to make money, have fun, and I think with biomimetics you can do the same thing.
Well, I'm a biologist, but I'd be the first to say that traditional engineering is probably a much, much more effective way to go in creating useful devices than evolution. Just look at cars, for example. They're actually astounding things. So I think what advantage we get from biomimetics of looking at nature is actually that we bypass the limits of human creativity. You don't have to just be creative. You can actually look at the solution that already exists in nature, and that sometimes is a solution that's completely non-intuitive. So I think that's something that's really important. You'll hear a lot of people say, don't copy nature, you should abstract the important stuff and then use it. That's true, but in my world, I'd also argue, don't throw away the complexity, because in many cases, I think the complexity of nature is actually the solution to the problem. So we have to try and understand the complexity better.
Well, the reason why I'm really excited about biomimetics is that I think it gives a great opportunity to better understand biology. So by building, for example, robots that closely mimic animals or plants or any other species, we can try hypotheses and measure things we cannot measure with real animals, or make modifications we normally cannot. I think that's really cool. At the same time, I maybe, because I'm both a biologist and an engineer, maybe a bit less enthusiastic about biomimetics as a method for design, where I definitely believe that I'm really a believer of where you look at the principles and see which principles make sense from an engineering point of view. And if you understand, for example, how an aircraft is designed, I'm an aerospace engineer, then you certainly understand that you don't want to copy how a bird wing is built up to the final detail, because it's so complex from so many aspects that if you would completely copy that, it would be, for example, much more well, it would be more dangerous, for example, because it would be less reliable. It could be much more expensive, for example, because of all the maintenance that might be required to some degree based on what we are currently capable of. But some of the principles are really useful, and they really help us to think out of the box, to get away from solutions that we have been trying to perfect for years and years and years, which is also a tradition in aerospace engineering. So I really see it as an inspiration for better understanding how, for example, flight works, and then applying the principles that seem to best match our current possibilities in engineering to improve our designs and fly more efficiently. Thank you.
Yeah, I mean, most has been said. Evolution didn't invent a bicycle or things like that, but with what it has, it finds very creative solutions, and that's helping a lot. And it has very huge noise, still can very nicely integrate information processing. And I see more and more that evolutionary techniques in design, say in a computer, is improving it. For example, lens design, people are now optimizing really from scratch quite complex lenses, partially from evolutionary dynamics, partially also from an engineering standpoint. So I think it's that you're not afraid from the complexity. That's something what you can learn from nature.
So I think I would like to just amplify everything that Barry said. He probably said everything I want to say, but I'm going to say it in slightly different words. I think many people here agree. Tony, you hit the nail on the head. The two directions are inspiration for understanding nature, from the machines we built as models to help us clarify our thinking, so we understand the biology better from biomimetics, and we can occasionally arrive at new technologies when we look and study systems which embody abilities that our technologies cannot today. And I think that's the two directions where biomimetics work. And the evolutionary arguments are exactly on point. Biological systems are inherently multi-constraint systems. They satisfy many constraints at one time. And so they're not going to be optimal for any one function that we might want to imitate. And the example of flight, I think, is a very good one, because birds evolved flight long before humans perhaps even imagined it. When we built human jets, when we built airplanes, we could exceed the capabilities of birds in terms of payloads and speeds. We can do things by optimizing that birds just can't do. But birds still have a few tricks to teach us, because they're far more maneuverable and capable of doing things that we would like our aircraft to do. We can still learn those tricks because we don't know how to do them by studying nature and doing it in a principled way.
Hi, so I speak as a engineer and designer, and when I talk to my students, my point is that bio-inspired design is one of these tools that you can have in your toolbox. And I don't think I have much to add to what's already been said, but I'd like to just build upon a point that was kind of brought up by both Barry Trimmer and David Lentink. So when we're trying to optimize for a specific set of conditions, we can often do very much better in bicycles, gas turbines, rockets, and so on. Part, I think, though, of what comes out in biological systems, because they are trying to satisfy many different conditions, some of which we know and some of which we haven't guessed yet. I think one of the byproducts of that is robustness, very robust interaction with the environment and with unexpected changes in that environment.
Okay, thank you, everybody. I just want to make one comment, and then maybe if people from the floor want to comment, they could put their hands up. Have we got another microphone? So, oh, good, okay. My comment was really to mention the second element of the conference, which is biomimetic and biohybrid systems. And biohybrid is a bit more of a new thing and a new field, but I think we've already seen some really interesting examples of biohybrid today, particularly thinking of Joe's talk and this idea that you might use living muscle as part of a robot. And of course, biomimetics is going to be an essential part of biohybrid systems, because in order to use a muscle, you're going to have to understand what kind of signals a muscle needs in order to operate. So some parts of the engineered mechanism are probably going to have to be biomimetic. So I think we are hoping that with this conference, and hopefully if there are future editions, we can look more at this biohybrid system aspect of it. And of course, in a way, everything we do is a biohybrid system because we're building robots or engineering other artifacts to be used by people. And so they are part of a biohybrid system in which we should remember the people that are using them. Any comments from the floor? Yeah, at the back. Oh, Nathan first.
So I suppose what I want to say is biomedics seems to me to be different from many other areas of science, in that science you often don't know the solution before you come up with the answer. Say, you know, quantum mechanics and so forth, they sort of came out of the blue. Whereas in using biomedics to inspire new technologies, we know the solutions already because we can see them around us, but the difficulty is actually reaching there. So I see biomedics as really given existence proofs of technologies that are possible in the future. And why is nature so good at producing these solutions? Well, we've had billions of years of evolution. And what is evolution itself? But it's a process, an optimization process of solving problems in the natural world. So that's why evolution, the natural world, has furnished all these problems to technological solutions that we can copy and exploit in man-made technologies.
I think there's a comment at the back. Could I just say that if you do want to say something, please just tell us who you are first so we can get to know each other.
Yeah. Okay, Holger Krab, bioengineering.
Well, we can come back in a second. Yeah.
Holger Krab, bioengineering. I work in insect sensory motor control. So I think the list was pretty complete, which the panel presented, but there's one constraint I would like to emphasize in terms of biological systems and systems design, in fact, and that is energy efficiency. So I think evolution had always one of the major constraints, which is you do not want to waste energy. And that is actually true not only in terms of, burning calories for movement, but it also goes into the area of sensory processing, which is made particularly efficient as well. So nervous systems operate under energy constraints. And then, of course, if they don't get it right, then the motor system will waste loads of energy. And that is one of the major driving forces, I think. So just as one addition to many of those complete.
Any more comments from the floor? Yeah.
Yeah. My name is Steen Rasmussen. I'm a physicist from Denmark. So we work on similar things. I mean, I would formulate it in a different way. I would call it living technology, where the emphasis, and I come from the artificial life field, and I would emphasize the desire or the goal is to make living or lifelike processes in all kinds of different materials because of the inherent power that living systems have and have shown, but not limit ourselves to look after what nature has already done. And this has been emphasized several places, but I think that, you know, I haven't seen everything here today, but it seems like this is in the spirit we have here. So it's not just, I just feel that the name biomimetic is limited. I would rather like to see that we explore life or these open-ended, innovative processes in all kinds of ways as a different design principle in engineering.
It's Pablo Barona. I like to emphasize, one of the properties of biology that for me is most powerful is the self-regulatory mechanisms that the biology has. And in my opinion, this has to do with the information processing at very different spatial and temporal scales. So all the relationships that living organisms have at the going from the molecular level all the way to the whole system level. And this is so far from any engineering device that I think well, it has to attract the attention of most of us.
Okay, there were some responses. So
I'm going to try and keep it brief so the discussion's open. I agree with pretty much everything that's been said. There are some riders to this. For example, the energy efficiency, that's true for most of the animals that we traditionally think about, but there are some animals that are not energy limited. They essentially live inside their fuel source, and for those guys it's going to be a very, very different set of conditions. So I think we have to be very, very careful about which model species we're thinking about.
So I would just pick up on that idea of existence proofs. I think that's a very important concept to emphasize, that when we're looking for places to put our effort in biomimetics or bioprinciple work, we should look for existence proofs of processes that we do not yet understand. And that's the great thing. When an animal does something, we don't understand how it's doing it, then we know we have something to learn, and that is where the science comes to bear. That's the thing to emphasize.
Yeah, okay, I'll be quick. One of my observations is that in biomimesis anyway, it's actually very hard to copy exactly what nature does, because we don't have, in many cases, for example, the material science that allows us to do the things that nature does. And you know, we don't make, in my world, we don't make dolphin clicks using biological structures. We use transducers and stuff. What we do is copy what they're doing in the clicks. The other example, so that's one observation. The other thing is, you know, sometimes the solution to what the problem you're studying isn't what you think it is. And the example that we came up with earlier was flying machines. We've all seen the early examples of flying machines from the turn of the 20th century, over 100 years ago. And there's these structures with wings that are flapping and umbrellas going up and down, and it's all unstable and the whole thing's falling apart. When the Wright brothers finally figured out how to fly, the problem they really solved was a control problem. Right? How do I do wing warping, as it turned out to be, so that I can make the thing fly straight and level and keep it, you know, and once they'd understood what an aerofoil was. So they had a lot of insight from animals and from birds in particular, but they didn't solve it by doing what birds do. They had a different approach, and it was control was the thing in their world. That was the problem they had to solve. So my take on it is, it's good to study biomimetic and biomimetic topics, but what you actually end up building that's going to be useful might be something slightly different than the thing that you're searching for in the first place.
I think that's a very good point, which fits with the next question. So what I put down to discuss next was we put a definition of biomimetics in the proceedings. We said biomimetics is the development of novel technologies through the distillation of principles from the study of biological systems. And so the next set of questions is one, do we like this definition? Do we agree with it? If so, what are design principles and how do we identify them? And is there a danger of circularity here, where we just call as a design principle the thing that we thought was useful to copy and that worked? So I'd like comments on any of those three questions. I think from the panel first, but I don't think we need to go in order, or everyone needs to speak, and then also from the floor. So would someone from the panel like to comment on this definition and what they think design principles are?
Thank you. Yeah, I would worry about the word design principles. And the reason is that I think design principles is very much related to the way we actually change and shape our world. Whereas in nature, it's more maybe governed by physical processes, and also, well, some of the work, like for example with genetic programming that we really don't understand that well from the design perspective. And as an engineer, I often think that whenever I start to use design, and I need to use that everywhere, it's because I don't fully understand each and every aspect of the system, and I need to make an informed trade-off. And I'm sure this is something that can be debated, and I look forward to hearing others think about it, but definitely would be careful with using the word design principle when thinking about biology.
Related to that, I often like to make the distinction between bio-inspired and bio-informed. We see a lot of devices that are inspired by the capabilities of animals, but not necessarily using the same concepts, principles, or information gleaned from the animal. Slightly more controversial, I think, is that we might want to think about dividing biomimetics separately from actually using biology in our engineering. I think those two are slightly different things. They overlap. But if you're actually using living tissue in your devices, that might not be just biomimetics. I'm not sure what you'd call it, but we should. Biohybrid. Biohybrid, maybe. Yep. Yep.
So I think the definition is fine as it is, but it does sort of bias towards the direction of developing new technologies and not closing the loop back to the biology. And my emphasis has generally not been on developing new technologies, but using robotic systems as a way of evaluating the hypotheses of the biology that I'm interested in. And I think there's an important loop to be had there to avoid the circularity that you're worried about. Because if we build a robot or some device or some apparatus, and we use the biological system as a yardstick for judging how well we've done, we're evaluating our theory. But then we can feed back from that to inspire more biological investigations, and we can cycle back and forth and converge on the truth of the mechanism, kind of going to what David said about we'll be surprised by what we find when we start going down that road. Enough iterations between the biology and the engineering informing one another maybe get us to that new principle, which is a new technology.
So Tosha, I think, was...
If you say biomimetic engineering, it's fine. That's very good, okay? So if you say biomimetic engineering is the development of novel technology, that's very correct, I guess. But if we say biomimetics, then biomimetics I should define in such a way it's a kind of science technology in the multi-scale from the kind of nano to the kind of macro level to discover, to understand the nature and also discover some new and also can make use of those such a kind of principles. I think this is a kind of a sort of principle. So that one example is like biomimetics can give such a good tool to the biologists to discover new principles, for example, or in bio or medical area too. That's why we are emphasizing such a biomimetic approach. That's a kind of science technology in the multi-scale in a sense.
Yeah, it seems to me that this description, it seems okay, but speaking as an engineer, I certainly recognize it as a very engineering way of looking at things in terms of was said already as a one-directional transfer of a more traditional engineering approach. Whereas I think we need to be placed much more in the center between science and a traditional sort of translational engineering design concept, in the sense of maybe thinking more about mechanisms and processes, as was talked about earlier, and really embedding ourselves within the interaction of these processes to maybe understand in a sort of pre-design stage, really, that maybe this sort of definition can then come after that, But in order to be able to get to this point, we really need to be able to understand what we're dealing with, and that's much more of a scientific process than an engineering process that is suggested here.
I'd like to distinguish between mechanisms and models, because a biological mechanism ultimately has to be able to derive from DNA. A model can be a set of equations, and unless you can specify biologically how the molecules and cells get together to produce a process, you don't really understand the mechanism. You're just modeling it.
Any comments from the floor? Yeah, the back there.
I'm Tim Tim Landgraf from Berlin. I'm working with honeybees, so I'm actually working with animals, and I want to use robots to find out about animals. So that definition is not what I would, sign up for when I'm coming to a biomimetics conference. So I think the definition should be as broad as possible, because we are, rather broad, group of people from different, fields.